All right, ladies and gentlemen, welcome back. We are going to continue our discussion on classification and really start layering in that molecular data, that DNA, that RNA, that protein information. Because remember, we talked about the fact that DNA exists in your nucleus, right? That information is what's passed on from your parents, it's your set of instructions. Your cells then use that DNA to create RNA sequences, RNA chunks just for the information they need to express that day, and then that RNA goes out to your cytoplasm, to those ribosomes, and that's where we build the proteins. And the proteins are really what we see in terms of cellular structure, cellular function. Those enzymes are proteins, for example, right? So those proteins are really what gives your body its characteristics. So we can look at differences in those sequences, whether they're DNA, RNA, or protein, as a way to say, well, if these organisms have lots of differences between them for this particular protein, that means they must be really distantly related. If they have lots of the protein in common and there aren't a whole lot of differences, then they must be much more closely related. So we're going to use that molecular information on top of the physical characteristics, the things that we can physically see, fur color, height, what is their job in their environment, that kind of thing. And that's what we're talking about when we talk about cladistics. So cladistics is just the classification of organisms based on common ancestry. And we create cladograms, which is what this is. This is a cladogram. And these are just evolutionary trees. They are hypotheses, they are proposed explanations of how closely related certain organisms are to each other, and they're based on these things called derived characters. And those derived characters are traits that we use to figure out evolutionary relationships. So those derived characters can be physical things. Do they have feathers? Do they give live birth? Do they swim? But those derived characters can also be the molecular stuff, the DNA, the RNA, the protein. So when we look at these cladograms, right, these are the different species at the top that we are trying to study. So in this case, we're just going to call them organism A, B, C, and D. Along the sides there are the different derived characters that I'm using as a way to classify those species, to put them in this particular order. So presumably trait 1 would be something that all of these organisms, A through D, all have in common. That's why we can connect them. Trait 2 would be something that B, C, and D have in common, but A doesn't have. Trait 3 would be something that only C and D have, neither A or B contain, and then trait 4 is just something that D has. And so what we do is we create these branches. So each of those branches breaks off when that species no longer has that trait in common with everybody above it. And so when we look at the branching points, those are the common ancestors. So we would say that when we look at this lineage, right, lineage line, evolutionary history, this is the common ancestry where A diverged from the rest of them. Here's a branch point where B broke off and started evolving itself, and C and D kept going. Here's the branch point where C broke off and D kept going. So these are their common ancestors. Doesn't mean that they are identical to the current species that exist. It just means that that's where we diverged from each other. That's the divergent evolution that we talked about. So what we create are these clades, or groups of organisms that share certain characteristics that are common to their common ancestor. So when I look, I can have multiple clades nestled into bigger ones. If you've ever seen the Russian nesting dolls, that's kind of what this reminds me of. So all of these organisms would be that biggest doll. All right, I go into that big doll and I get to a smaller one. That's common to all of these guys, so on and so forth until I get to the smallest doll in the middle. And now those traits are only connected to that smallest set of organisms. So each of these branch points or these intersections are where that common ancestry exists. We call those the nodes. And then the derived characters are the ones that are here at the hash marks. And again, these can be physical characteristics like you see here, the forelimbs, the embryo, the skulls, or this can be just numbers of differences in their molecular sequences, whether that's DNA sequence, RNA sequence, or protein sequence. So we're creating phylogenies, we're creating hypotheses for their evolutionary history for a certain group of species. How far back do I need to go before I can find a connection based on the traits that I'm observing for a set of characteristics? If we're specifically looking at molecular data, right, so the nucleotides, the RNA, the proteins, that's then specifically a form of phylogeny, a form of cladistics called molecular systematics. So cladistics is a bigger field where I can be using all sorts of data, physical data, um, ecological niche data, molecular data, it all fits within cladistics. If I want to go specific and talk about molecular information, molecules, DNA, proteins, RNA, then I'm nestled within that field of cladistics, specifically looking at molecular systematics. You may also come across the term phylogenetics or phylogenics, where they drop that sort of middle here. And those phylogenics, it's the same thing. We're looking at phylogenies based on genetics. That's where that term comes from. And so we can create just traditional cladograms, which can include all sorts of data, molecular, physical, or otherwise. If it's specifically um, molecular data, then you may come across this term phylogenetic or phylogenetic tree. Um, Again, a lot of these terms are being used interchangeably anymore because so much of our data is based on genetics. Um, but I just want to throw all of that out there to you in case you come across one term versus another. So it's not necessarily inappropriate to call a evolutionary tree a cladogram, um, but if it's based on molecular data, then you may also be able to use the term phylogenetic. So just be aware. They've all got the same basic branching patterns. They all have the same basic, we use derived characters to build them. The species go at the top based on how many things they have in common. The construction of it is essentially the same. It's just what kind of data are you using to build the tree. So this is just, again, another example of what we're looking at. So again, all of this, they're calling it a phylogenetic tree, but realistically, this is all physical data. So it's a cladogram that probably is backed up by genetic information. Um, so again, those terms tend to get used interchangeably. The big thing is, can you read them? Can you read them? And then we'll also practice building them. So this is going to be a really great slide to constantly come back to as a reference. So let's talk through what we've got here. So if I look at that phylogenetic tree or that cladogram that I'm building, we talk about it having a root, just like a tree, the base of the tree. That's the common ancestry to every branch that comes off of it. So every species that is at the top, its common ancestors at the bottom. And then as I move up that tree and start seeing those branches, I'm moving closer to current times. So the deeper in that tree, the closer to the root, the further back in time I have to go to find that common ancestor. The more I get towards the top of the tree, the closer in recent history we're moving. Each place where that branch breaks, where that lineage splits, right? So here's one, here's another, here's another. That's a speciation event, right? And remember, speciation is the creation of new species from something that already existed. So that branch point means that something has changed. Something in the evolutionary histories of the species that are going to come from that branch has adjusted. They've moved into new environments, a mutation has occurred, some new characteristic has come along that now separates them from others. So if I look at this branch point here, that means that something changed to send species 2 off to look by itself, where species 3 and 4 continued to stay on the same path until they also experienced a speciation event and split off from each other there. If I look at the lines here, these are shared versus unique histories. So any point where that species is by itself, that is unique history to that organism. That doesn't mean that what they look like here is identical to what they look like now. It just means that they've branched off and their evolutionary history from that point forward was unique to them. They didn't share it with any other organism in the tree that you're building. If we have those connections, then those points are shared history between those species until they branch off by themselves. And again, each branch point then is where they share that common ancestry. Any place where it's unique history to them, that's their own unique ancestral lines until you get back to the branch point itself.